I'm pleased to announce Dr. Emma Hodcroft as the next speaker. Emma is postdoc and researcher at the Institute of Social and Preventive Medicine at the University of Bern in Switzerland, specialized on HIV simulations and on open access policies. She's been one of the clearest and most engaged voices during the pandemic, explaining the virus and its functioning to a broad public in a very strong way. And we thought it would be important to have this voice also as part of engadinarttalks. I like what she has posted on her website. It's a quote by Isaac Asimov. The most exciting phrase to hear in science, the one that heralds discoveries, is not Heureka, but that's funny. Please welcome Emma Hodcroft. Thank you.
Hi, my name is Emma Hodcroft, and I'm a researcher at the University of Bern. And I'm really pleased to be here today and have the opportunity to talk to you. And today in particular, I'd like to focus on human-virus interactions over weeks and centuries. Now, I think particularly this year, we've had a lot of time to think about time. And in particular, time in a pandemic has been a somewhat fluid concept. Some months have felt like they've lasted for years. Other months have flown by in a second. And I think it's been a great time to reflect on how our activities, our restrictions, and how what we're doing every day changes our perceptions of time. But today I wanted to focus particularly on talking about viruses and immunity in 4 different scales of time. So in particular, I wanted to focus on acute infection, so about 2 to 3 weeks for most viruses. So how do we fight infection and how does immunity work? And then I wanted to talk about, of course, very timely, time in a pandemic sense. So maybe 1 or 2 years. And in particular, I wanted to talk about some of the hot topics that you'll hear in the news today, like how do vaccines work? And do we have some fears about whether the vaccines will work right now? And then I wanted to move to kind of a human lifetime scale. So maybe around 80 years, thinking about, how long does immunity last once we've developed it? And how do viruses respond? What do they think of this? And then finally talking a little bit longer than human lifetimes on the scales of centuries. How do human immunity and viruses impact each other? So to start, let's talk about acute infection. So kind of how does immunity work? A little bit of a brief primer on viruses and immunity. So I like to think of viruses as kind of infectious instruction manuals. They can't survive on their own. They need host cells to replicate. They essentially just have enough tools to get into a cell and give it an instruction manual for how to make more viruses. But of course, this causes the host cell to die, sometimes directly in that it makes so many viruses and then they burst out of the cell, killing it. But also indirectly, cells will commit suicide if they feel like they've been infected, or they'll flag to the body to come along and kill them. Because of course, we don't want our cells to be virus factories. We have other jobs for them to be busy doing. Now, initially, when we're infected by a virus, we don't recognize the virus specifically if we've never seen it before. But our bodies make what you can think of as kind of a generalized response. We make fever, we might increase mucus production, and we make chemicals like interferon that help against lots of different pathogens, and particularly against viruses. And this is our body's first defense, to just act like something is wrong, a pathogen has invaded, and these are techniques that seem to work. But over time in the infection, we will learn to recognize that virus. We'll recognize bits of the virus, and in particular, usually the bits that are on the outside of the virus. In coronavirus, these are called spikes. You'll have heard about them in the news. They're the most sticky out parts of the virus, so they're kind of the easiest parts for our bodies to see. And we develop what are called antibodies that match these bits. They can recognize those parts. And these stop the viruses in 2 ways. First, they attach to the viruses and kind of surround them. And if they're surrounded by antibodies, they can't touch a cell, which they need to do to infect it and to replicate. They also flag our immune system. They're kind of a wave saying, hey, I've got a bad guy over here, come and get him. And our immune system cells will go over and eat that virus up and get rid of it. And between these 2 responses, our generalized response, which in biology we call the innate response, and our adaptive response, or the antibodies, we can clear the virus infection and get healthy again. But why does developing antibodies matter? What do they do? Well, it's a question of next time. If we have antibodies and we see that virus again, we can recognize it more quickly and we can get a really good response going more quickly. Ideally, that means we don't even get infected at all. But in other cases, we might get infected, but because we can keep the virus in check really well, we might not get as sick, we might not have as severe symptoms, and the duration might be shorter. Now, just like how we learn to recognize our friends and our family, we don't just learn to recognize their left eyebrow. We learn to recognize many different parts of their faces, and antibodies work the same too. We learn to recognize different parts of the outsides of the virus and even some of the insides as well. And so with that, we can recognize viruses pretty well. But certainly this can change over longer timescales. So if we move to thinking about a pandemic timescale, maybe 1 or 2 years, we've certainly got 1 year behind us, certainly hopefully a small number of years, a lot of questions have come up. So how do vaccines work? Are they different from that natural immune response? Are there fears about whether the vaccines will work with new variants that we're seeing? So first, let's just cover quickly, how do vaccines work? So vaccines, if we think about viruses as kind of infectious instruction manuals, mRNA vaccines are essentially instruction manuals, but with almost all the pages torn out. So they only have enough in them to kind of go to a cell and hand them 1 page of an instruction manual, how to make those sticky out parts of the cell. So in coronavirus, these are those spike proteins, the outside parts of the cell. They aren't able to make a fully functional virus that could go on and infect other cells. They just have one job, hand over the instructions to make the spikes. But these serve the same function. They go out into the body and we learn to recognize them so that if we see those spikes again, this time attached to a virus, we can make a fast and effective response. But of course, the question of the moment is, what if the virus changes its spikes? So here I've just changed them from green to red. And what does that mean? Will our antibodies still work? Will we still be able to fight the virus as effectively? Now, one thing that's helpful here is that antibodies aren't perfect, and that's a good thing. It's just like how we recognize people, the same analogy I used earlier. If I showed you this picture of this woman, even if she changes her uniform, is in a slightly different light, and pulls her hair back, you could still say with a lot of confidence that yes, this is the same person. And even if she's very responsible and she goes out shopping with a mask on, you could probably still recognize her in this photo, that it's the same woman. Now, this is exactly the same as how it works with antibodies. Not only do they learn to recognize many different parts of the virus, but they're also not perfect. So even if the virus changes a little bit, many of them will still work. Maybe not always as effectively, but effectively enough that they can prevent infection or prevent severe disease. And so this is what we hope, is that between both the fact that the virus recognizes many bits, the antibodies recognize many bits, including the parts that haven't changed, and the fact that those antibodies are not, perfectly bound to just one very, very specific thing, this means that we can recognize we have a better chance of maintaining immunity, even if the virus changes a little bit, just like the woman changes in these photos. Now, of course, the worry over longer timescales is that, of course, there is different amounts of change. So if this woman went on and, as well as the mask, the uniform, she puts on a hat and sunglasses, now we might be a lot less confident that this is the same woman. And of course, this is the concern with the virus as well, that if it changes enough, our bodies might not be able to recognize it. And so currently, we've had some good news this week about the newer variants that are circulating of SARS-CoV-2, that it seems like the vaccines can still work against these variants. So our bodies, after having the vaccine, can still recognize these variants. So we're kind of about at maybe picture 2 here, where the virus has changed, but our bodies are still very effective at recognizing it. But the future worry, of course, is that if the viruses change enough, if they put that hat and sunglasses on, we might not be able to recognize them from what the vaccine has trained our body before. The good news is, though, we can update our mugshot. So just like if you knew that this woman was now going around with sunglasses and hat, you could start showing people this picture if you wanted to identify her. Well, it's the same here. We can update those vaccines so that they're showing our bodies the red spikes instead of the green ones, and we can stay updated to the most current circulating forms of the virus. Now I want to move to a longer time scale, though, because even when we've made all of these antibodies, what does that mean longer term? On a human lifetime scale, how long does immunity last, and how do viruses respond to this? So, once we've made these antibodies, what does it mean for the longer time of our lives? So once we make these good antibodies, hopefully the kind of idea is we're protected, right? I mean, we see something, we make antibodies, and we're good. I want to move now to talking about a slightly different virus, enterovirus D68. This is actually the virus I studied before the pandemic. It's one I'm very fond of. It was discovered in 1962, and it's primarily diagnosed in young children. It causes mild respiratory illness, so fever and cough. And it does seem like pretty much everyone gets this virus when they're a kid. If we look for antibodies against the virus in the general population, pretty much everyone has them by age 17. And so for a long time, the general thinking was that essentially you get this as a kid, you develop really good antibodies, and when we test these antibodies, they seem to work really well. And then you never get it again. So it just only infects young children who just haven't seen it before. It circulates in young children, and that's about it. But we can ask ourselves, well, do these antibodies work that well? Do they work well forever, that you never get infected again? And we can actually test this. So one of the ways we can test this is we can take that earliest enterovirus that we found, that earliest D68 that we found in 1962, and we can show that to the antibodies of someone who was born at around the same time, who probably saw this virus as a kid. And when we do, this guy, Henri will call him, he recognizes that virus so well. His antibodies do a great job. They are all over that virus. I remember you, let's get a response going. But if we take a more recent virus, say one from 2014, and we show this to Henri's antibodies, they do still recognize it, but not nearly as strongly. He still has a pretty good response. He's not going to get very sick from this, but it's very interesting to note that he doesn't recognize it as well. So what has happened here? Well, two things. First, the virus has changed over the last 60 years. We can actually see this in the genetic material of the virus. Now, if the virus only infected young children, it wouldn't have to change because they don't have any immunity. So there's no reason to change. You're working good just the way you are. But the virus is changing because it's trying to get around immunity. And this most likely means that it is infecting adults. If it was just infecting children, it wouldn't have to change. The fact that it's infecting adults means that it is changing. And so this idea that adults don't get D68 might not be true. The next thing that's changing is the next thing that might be happening is what we call original antigenic sin. This essentially means that Henri's immune system, it's stuck in 1962. That was the first virus that he saw from enterovirus D68, and it's still the one that he's thinking about in his immune system. So he'll recognize that one the best. Even though he will have seen many other D68s come and go since then, he thinks the original is best, and he hasn't really updated his mugshot, we might say. He hasn't learned to recognize newer viruses quite as well. Between these two things, one interesting thing that this indicates is that adults might actually be a reservoir for D68. They're where the virus kind of hangs on in the corners until it can get into children, where it then spreads very happily through schools and nurseries and brings home the many colds that we know that children often do. And so this really shows us how that interplay between viruses and humans can change over a lifetime and impacts how the viruses are evolving as well. But let's take a look at that on one more zoom out larger time scale. So on the course of centuries, how does human immunity impact viruses on longer time scales? And here I'm going to switch viruses again to talk about influenza. Now, it's actually pretty difficult to study viruses over long periods because we didn't have tests to distinguish them, of course, and the symptoms are really similar. Historically, people just write down, everyone had a fever and a cough, but that describes a lot of respiratory viruses. And even if we can be confident, for example, that it was influenza, we don't know which strain or which type it was. And so we're often limited in how far we can go back. And of course, the tests we have to distinguish these are relatively recent inventions. But we do have some longer term data on influenza. And that's what I want to show you now to talk about that longer time period. Now, I think one question that really illustrates this is if everyone's had flu, regularly in their lives for hundreds of years, how is it even still around if we all have some immunity against it? And the answer here is really that influenza evolution is driven by human immunity. We're in an endless game of cat and mouse trying to catch each other and trying to stop each other. So flu has a nice ability, well, it thinks it's nice, to mutate relatively quickly. So faster than enterovirus, faster than coronavirus. And it's under constant pressure to evade the immunity of the flu that was circulating last year and the year before. And because it mutates quickly, it can actually respond to that pressure. And we can see this in the genetic material of the flu. So this tree that I'm showing you now is the sequences of influenza from the 1980s until 2015. And what we do is we've plotted them by the date that they were taken on the x-axis. And then we see how similar 1 year's flu are to the next year. And this is a very distinctive picture. It's called a ladder-like tree, but I always think it looks a little bit more like stairs. And you can see that year on year, influenza has tried to move away from what was circulating last year so that it can get away from what your body will recognize and be in a position where it can infect you, even if you might have been infected last year or the year before. And this drives this very linear stair-step type evolution over time. And we can look at this in one more way, but to do that, I just have to tell you a little bit more detail about flu. So flu also has these outside sticky out bits, like the spikes in corona. These are called HA and NA. There's actually two types on the outside of flu. And we aren't very creative in science. And so there's many different types of these HA and NAs, and we just name them by numbers. So we have 16 types of HA and 9 types of NA. And then we describe flu with things like H3N2. You might have heard this before, or H1N1. And this can tell us a little bit about, which flu we're talking about. But also, because these are the parts our bodies recognize, they can give us some information about how well flu might recognize. If you've been infected with H1N1, you'll also probably recognize H1s and N1s in other places, in other types of flu. So we can go back just for a minute to something else that's been talked about a lot in the last year, the 1918 flu pandemic. We aren't really sure what type of flu was circulating before this, but we do know that that was an H1N1 type of flu that jumped into humans, we think possibly from birds, in 1918. It must have been different from what was circulating previously because it spread so effectively and of course was so incredibly damaging and caused so many deaths around the world. And it became the dominant flu strain. It jumped into people, it spread incredibly quickly, and then it kept circulating. Now, because people had seen it after a few years, it wasn't as deadly, but it was able to keep on circulating. Now, interestingly, you'll see, well, we'll talk about this in just a second. There's a little gap here that I'll cover exactly what happened. But the next event that happened is that in 1957, a new version of flu, so this is H2N2, jumped into humans. This one, again, it took over from the circulating H1N1. It spread through the population, thankfully wasn't quite as devastating as H1N1, but it also then began circulating. It was that big outbreak and then changed into a seasonal pattern as people developed more immunity. It didn't get to circulate for too long, however. In 1968, a new version of flu, this time H3N2, also jumped into humans. It overtook the 1957 version, and it then became the circulating version of flu since then. Now, we also had a reappearance of H1N1 in the 70s. Interestingly enough, this seems to be a freezer escape. So H1N1 was made extinct by the 1957, but there were samples in a freezer somewhere that got out and got back circulating into the population. We can actually see this because we can see that there's no evolutionary change between the 1950s and 1970s. And that's just not possible if the virus was circulating. So it must have literally been frozen and then gotten back out. But it only got to circulate for a few decades because in 2009, as many of you will remember, we had the swine flu outbreak or the pandemic H1N1, a different type of H1N1 that jumped from swine into humans in 2019. Again, thankfully, it wasn't so devastating in deaths, but it did spread extremely well. It knocked out that resurrected version of H1N1, and now we currently have H3N2 and H1N1 circulating together. But what we can see here is that because if you can evade that human immunity, if you can be different enough, you have a chance to jump in, knock out everyone else. You are then the circulating variant, but with every year, people become more immune to you. And so there's always a chance that a more divergent strain, a new version of flu might jump in and knock you out by spreading so wildly. And we can really see clearly how this pandemic H1N1 knocked out the original H1N1 in this chart. This is just number of flu cases in the US year on year. You can see its seasonal pattern. These are winters. And here the original H1N1 is in blue. The pandemic H1N1 is orange. This is the 2019 pandemic. And after this, you can see there's no more blue. So it has knocked out blue and taken advantage, all in response to what human immunity allows the infections to happen more easily. So I hope that this talk has given a little bit of an interesting overview and a tour through the different ways of thinking about viruses and human interaction and how that works on different timescales. And I think the main takeaway message here today is kind of to think about this amazing feat that what happens at a microscopic level in just a few weeks with creating antibodies has this incredible long-term impact that shapes viruses over decades and centuries and causes this kind of long-term dance between humans and viruses, not only in our shorter infections, but also in this long-term scale of years, of decades, and of centuries. And with that, I'd like to thank you very much again for this opportunity to speak, and I hope that this has been an interesting and educational talk. Thank you very much.